take, I'll start with attendance. Hoping more people show up and announcements. So, if I call you, well, first of all, who has the big issue here? You two? Okay. If I call your name, that means you're not here. So if I call your name, let me know. I'll fix it. Billie Jean Davis, Tell, Sims, Fisher, Whitaker, Stanley, Newton, Davis, Smith. Sorry about that. Davis, yeah, thanks. Sorry. First person here, I'm not talking to you, and still. That's it. Anyway, Smith, Dunlap, and Carr. Anybody I missed? All right, I'll do some slow announcements. I hope more people show up. But Donovan was out of town this weekend, like I told you guys. Yes, sir. I got you. Yeah. Didn't call your name. You're good to go. So yeah, I had to travel this weekend. That was half the problem. The other half, if you look at this from the academic calendar, we are now halfway through the semester, which for you guys doesn't mean too much, but for me it means a lot because I teach an online course. So my first 8 weeks is over. So that's a lot of work, right? Getting everything graded and getting everything ready because that's the end of their semester. Likewise, the second 8-week course started on Monday. So that's a lot of work. So again, I just wanted to apologize to you guys for being a little slow to reply to emails and grade and things like that. So that is why. And on that note, really quickly, I hope this will be the last time I make these long announcements, because we are halfway through the semester. So at this point, you should know how things are going. Feel free to ask me if you don't, and come to me for help. But I'm going to start moving forward. I'm going to stop repeating myself so much because the people who get it, get it. The people who aren't paying attention just aren't paying attention. They're just not getting it. But one last time. So this is midterms, right? You should know what's going on by now. You should really take a hard look at yourself and figure out, can I finish this course and pass it? Because I'd have my grades in for midterms by Monday, this coming Monday, and then you have Friday to withdraw from the class. So take a good hard look at yourself and decide if you think you can really pass this class. I will say this, and this is historically, so don't go getting really cocky, but usually if you're passing by now, you're going to end up passing the class because you know what's going on. You've got that rhythm, right? And usually people don't screw it up. But again, if you're one of those people, don't just like keep shoes off and like, oh, I'm going to be fine. Usually people by now who are failing end up failing. But I'm telling you now, that's only because the people who are failing at this point, they just have other things going on in their life, and it's not, biology's not important. But if that's you think you can do it, I'm here to help. It's just going to take a lot of work. On that note, I'll give some advice again, and then hopefully I'll be giving less advice on these announcements, and we can talk more about biology. You guys need to do the bare minimum. And the first thing you need to do for bare minimum is pay attention. Pay attention. For example, how many of you will know, how many times, it's a rhetorical question, but how many times did I say verbally and in writing that the exam was going to be this Monday? Many times. Yet I had people email me and ask me when the exam was. That was you, don't take it personally. Let me preempt this. If you were one of those people, good on you. You individually, good on you, right? Because things change. So even though I said it many times, it does not hurt to write me and email me and say, hey, is the exam this Monday? Good on you for doing that. But as a whole, statistically, some of you who didn't email me and ask me was it Monday, were asking because you didn't know. I don't know if that's true, so I don't judge you individually. But pay attention. And again, like I keep posting announcements and say, if you read this, reply to it for extra credit. Some people still aren't just reading the announcements. That's part of the bare minimum. Read the announcements, come to class, and pay attention when I'm talking. I know it's boring stuff, but please do it. The other thing with the bare minimum is do the assignments, right? Do the independent work. It's basically 3 points, right? You just keep going until you hit 100. It doesn't matter how bad you screw up, you just turn in garbage. And I can write back, like, hey, that's garbage. It doesn't hurt you. Just keep turning in stuff, right? Learn from your mistakes, keep turning it in until you hit 100. And as some of you are already doing, you can go above 100. Get extra credit to help make up for your weaker points. Some people just aren't good test takers, for example. So that helps. Oh yeah, speaking of testing, sorry, let me go off on a tangent. There's still at least one person who needs to take the exam. This is why I'm not giving you feedback on the exam yet, but we'll come back to that later. So yeah, pay attention, come to class, either online or in person, and watch the videos. Do the independent work, do labs. A lot of people just aren't even doing labs. Even doing a bad job at lab is better than doing nothing. So do that. That's the bare minimum. You want to take it up a notch? Fix your mistakes, right? So for all labs, go back. If you screwed something up, fix it. Exam one. For weeks, I've said you could redo exam one. Only a few of you have done it. If you get a lower grade on your retake, then you still stick with your higher grade. If you get a higher grade, your grade is boosted. Read the textbook. Do the study guides, right? Those are all extra things you can do. So, I don't know. Like I said, we are halfway through. So, that's my last plea for you guys to either drop the class if you need it or come to me for help. And then one last reminder, if you come to me at our office hours and say, hey, let's talk about what I'm doing good, what I'm doing bad, how I can improve, you get extra credit points just for showing up, and we can push you in the right direction if you need it. It might be a really quick conversation. I'm like, you're doing great. Maybe you should help your classmates. Anyway, any questions, not about biology subject, but biology course. Anything about how things are going, do what I can do. If there's things I can do to make things better, let me know. I'm here to teach you. All right, pretty easy. So again, that's it for the announcements. First word for attendance would be WBSU. So speaking of which, if you're wearing the WBSU gear and you want to stay back for a picture in the hallway afterwards for a little bit extra credit, let me know at the end of class. And if one of you guys who's not wearing it, okay, I was going to say, wants to take the picture for extra credit, there we go. So one last time, any questions? All right, I'll address the exam soon. Long story short, some people did mess it up, turned it in late, and I don't know if that was a glitch in the system, or they just didn't know that it had to be in at 8:50. The good news is, because of that, I just felt bad, and now everybody gets to redo the exam. I'll post that later, but everybody will get to redo the exam. The redo will be non-timed, just like the first one, or the first exam one. Redo. Non-timed and open book. Just take it, use your book, look up all the answers, and your overall final grade for exam 2 will be the average of your first grade and your second grade. But for those of you who are upset because you turned it in late and are going to lose 10 points, apologize for that. But the good news is, overall, you're going to do better, probably, because now you're going to have an open book test to bump your grade up. And you've helped the rest of the class by doing so. All right, that's it. I'm in this time. Any questions? All right. Let's get to it then. Back to chapter 8. Feels like it's been a really long time since we talked about it. So let me catch you up. All right. So this whole section, chapter 8, 10, 13, I'll say that. But most of what we're talking about now is cellular reproduction, right? So the last exam, for example, was about transferring energy, right? The whole thing is about energy, one of the properties of life. This is about reproduction, cellular reproduction. We talked about mitosis. There's a lot to say about that you need to know. But the most basic thing to say is that mitosis is a part of the whole cell cycle, right? There's interphase and then there's the mitotic phase. And then we said mitosis itself is broken down into 4 stages: prophase, metaphase, anaphase, telophase. You need to know that. You need to know that in that order. You need to know what happens in those different stages. And then we said, like, towards the end, we have cytokinesis, which is when the cell, at the end of telophase, you do have 2 different nuclei, right? Everything is separated. But until you have cytokinesis, you still have 1 cell. So then you go through cytokinesis and you have 2 cells, right? That's mitosis. We also said that mitosis is what we use for asexual reproduction. So like you guys right now are reproducing. Obviously you're not having sex, right? But you are going through mitosis right now. You're making new cells. So you need to know that. You need to know that mitosis starts with 1 cell and ends with 2. 2 genetically identical daughter cells. Right? That being said, then we got into meiosis, and I told you, remember, for the exam, a lot of it's going to be knowing the difference between meiosis and mitosis. So the first thing we talked about was homologous chromosomes. And to remind you, you guys have 46, well, you should. You should have 46 chromosomes. Some people don't. They might have 1 extra or fewer. We'll talk about that later. But yeah, the average human the normal human has 46 chromosomes, 23 pairs. Homologous chromosomes are one. It's a set of chromosomes that you get from each parent, right? So let's say chromosome number 8, for example. You got one from your mom, you got one from your dad, right? That's homologous chromosomes. They have the same genes, so wherever the gene is for bad knees, which doesn't exist, but let's say it did, if the gene for bad knees is on chromosome number 12, then you have, a version of that gene that you got from your mom on chromosome number 12, and then you got a different version on a different chromosome number 12 that you got from your dad. Those are homologous chromosomes. What else do we say? It's important. I don't want to go too deep because we've already talked about this. Then we talked about gametes. Gametes are sperm or egg, right? Basically. And if you haven't done the pre-lab yet, there's your answer. Sperm and egg. What else to say about that? Oh yeah, it's a good time to remind you. When you do the pre-labs, make sure your answers come from the introduction. Don't look it up. The pre-labs are almost like the questions on the videos. The pre-lab questions are basically to prove that you read the introduction. You need to read the introduction. Also, this is a 100 level course, so many times you're getting the Cliff Notes version. So what I'm teaching you in the introduction might be a lot easier and slightly different than what you're going to look up, or if you look it up. So use your introduction. Anyway, those where gametes are. Then we finally got into the process of meiosis. And the first thing we talked about in this process of meiosis was the differences. Because like I said on the exam, a lot of this is going to be contrasting the difference between mitosis and meiosis. Pull this up, we'll be all right. Yes, here we go. Finally caught up. So the first thing we said, we already said this is where we left off. One of the big differences between meiosis and mitosis is in meiosis we have something called, in meiosis the chromosome numbers are cut in half, right? So in mitosis, if you start with 46 chromosomes, you end with 46 chromosomes. In meiosis, that's not the case, right? You start with 46, you end with 23. The numbers are cut in half because, not that you need to know this much detail for the exam, but hopefully this helps you remember. We only go through one S phase, right, as usual, just like mitosis. So you duplicate everything once. In mitosis, you cut it in half, right? So you double it, and you cut it in half. That puts you back at the original number. In meiosis, you double it, you cut it in half, which puts you back at the original number. Then you cut it in half again, which puts you at half the number. Which again, to remind you, should make sense because you're trying to, in meiosis, the purpose is to make sperm and egg. And you want to have half the information because you want those things to come together and make a whole. So two halves make a whole. We left off with that, and we also talked about this. Crossing over is the second big difference between meiosis and mitosis. So what happens in crossing over is your homologous chromosomes come together. So let's say this is chromosome number 13. I'm just throwing that out there randomly. You have a version that you got from your mom and a version you got from your dad. Once they get close enough, they sometimes literally swap chunks, right? So you will have a mom chromosome with a dad chunk on it. And then likewise, a dad chromosome with a mom chunk on it. And that'll be very important later in the semester because this is very much where we get our sexual diversity. We'll talk about that later. I also wanted to point out that this is just one example. It doesn't always happen at that part. It doesn't always happen at all. Also, sometimes it doesn't happen once. Maybe twice it would happen in the same situation. Or maybe it's a big chunk or a smaller chunk. Right? It's always random. And that's where we left off. So the second word for attendance will be the number 13. So it took me 13 minutes to get to where we are finally talking about new information. I apologize.
Is that why siblings?
Yes, that's a great question. That's one of the reasons. That's a great question. If you didn't hear it online, she says, is this why siblings look similar but different? And yes, that is one of the reasons. Because you're all getting genes and chromosomes from your parents, especially if you have full siblings, like the same mom and same dad. You got chromosomes from your mom and your dad, they got chromosomes from your mom and dad. Later, we're going to talk about, later in this chapter, we're going to talk about how you didn't necessarily get the same one. So say, for example, chromosome number 7. You may have gotten chromosome number 7 from your mom. How do I put this? I don't even want to put it. We'll get back into it once I actually have the stuff in front of me and I can actually show you. Because if I just verbally describe it, it might be. But yes, this is one of the reasons that you guys look similar but different. And this will be very, very important in the semester moving forward. Some of these things, like you just leave it in that chapter, This is the concept that's going to be important. True. Chapters. So any questions about this? All right, here's some pictures. Now we're jumping into the process of meiosis. We've talked about the differences, some of the differences between meiosis and mitosis. Now we're going to actually talk about the process a little bit. It should look pretty, whoops. This part should look familiar. The interphase is still the same, right? Because we haven't gotten into the mitotic phase yet. This is still interphase. This is the other part of the cell cycle. Meaning, the centromeres are just sitting there, they're not doing anything yet. We know that they're going to be an important layer. More importantly, the nuclear envelope is still intact, right? The chromosomes are untangled, and we talked about why when we talked about mitosis. Then we get into prophase. And this, again, should look very familiar. Because remember, what we talk about in this course, what we're focused on is what happens to the chromosomes, right? And what's happening to the chromosomes is we're putting them together and then separating them into two different nuclei. So everything that happens, and this is going to be a refresher for what we talked about mitosis, should be logical. First thing we need to do is get rid of the nuclear envelope, right? Because we're trying to make two different nuclei on different sides of the cell. So we need to get rid of the one nuclear envelope that we have. That's gone. The chromosomes need to condense, right? We don't need them to be a big squiggly mess. We need them to be condensed so we can move them around in an organized fashion. Speaking of moving them around, we need to attach the things that's going to move them around, right? So this spindle attaches to the chromosomes. This is all in prophase. Here's where it gets slightly different. So if you take notes, maybe write this down. Because again, you will need to know the difference between meiosis and mitosis. In mitosis, we weren't talking about homologous chromosomes being together. All we were talking about was the sister chromatids. So it's meiosis 1 that deals with the homologous chromosome. We never even mentioned homologous chromosomes when we talked about mitosis. So if you have any questions about homologous chromosomes on the exam, the answer should have something to do with meiosis. Specifically meiosis 1. So if there's a choice between meiosis 1 and meiosis 2, the answer is meiosis 1. It will have nothing to do with mitosis because we're talking about homologous chromosomes. So anyway, as far as this picture is concerned, there's the one difference between meiosis and mitosis. So far, everything else is the same. So now we're talking about homologous chromosomes. We're moving homologous chromosomes around instead of sister chromatids. Don't get me wrong. Obviously, the sister chromatids make up the homologous chromosomes, so they're still there, but we're not talking about them yet. Then we move into metaphase. This is just the same as mitosis, right? What happens in metaphase? The chromosomes are in the middle, right? And there's a tug of war going on right now. We can't see it, but these things are pulling the chromosomes. Specifically, and again, this is how it's different than mitosis. It's pulling the homologous chromosomes, not the sister chromatids, right? It's pulling those homologous chromosomes, trying to separate them. That's metaphase. What's anaphase? Well, the tug of war finally did its job, and it separated the homologous chromosomes, not the sister chromatids, right? It separates them and starts pulling them towards the opposite ends of the cell, and then finally they make it to the opposite ends of the cell, and that is what we call telophase. So again, that's basically what happens in mitosis, except here we separated homologous chromosomes. We didn't separate sister chromatids. The sister chromatids are still together. We haven't done anything with those yet. Even though on the last discussion we had, I told you the difference between haploid and diploid cells, and even though there's a question about this on the study guide, I want to keep things slightly simple for the exam. So you don't need to know that by the end of meiosis 1, because remember there's 2 rounds of meiosis, by the end of meiosis 1, you end up with 2 haploid cells. But I'm not going to ask that on the exam. It is on the study guide, but it's not going to be on the exam. So that is the first round of mitosis. Next, we're going to do the second round of meiosis. Are there any questions? Yes, sir.
Can we do that for even this will be on the study guide?
Oh yeah, definitely do the study guide. Well, I shouldn't say
No, I'm saying
definitely.
even if it's in the study guide,
Yes. You mean write about that?
like for independent.
Yeah, you can write about it for independent work. It's a good time to remind you guys, if you guys write about something on independent work, the idea is for you to look at a source other than textbook. I want you to learn other stuff. I'm teaching you what you need to know from the textbook. I'm hoping you guys will find stuff that you either find interesting or that you need help with. Like I've read what the textbook said about meiosis 1. Now I want to go and get a different version, a different explanation. So great question, thank you. Any questions about meiosis 1? All right, meiosis 2. Long story short, meiosis 2 essentially is mitosis. Right? So we're still dealing with the same thing, right? We still need to attach the spindles so we can move the chromosomes and prophase. They still meet in the middle of metaphase and tug of war is happening. Once the tug of war finally does its job and they break, at the point that they break, that's still called anaphase. And they're moving towards the end of the cell. Once they finally get to the end of the cell and they reform the nuclear envelope and all that stuff, that is still telophase. And you'll notice this time in meiosis 2, it is the sister chromatids, right? We already separated the homologous chromosomes. Now we're just dealing with the sister chromatids, just like we did in mitosis. So as far as we're concerned, meiosis 2 is essentially mitosis. Of course, overall, the big picture, and I've said this before today, and I'll say it again, mitosis, you start with one diploid cell, you end up with 2 genetically identical diploid cells. In meiosis, you end up with 1 diploid cell. Oh, I don't want to get a picture of it. No, anyway, and you end up with 4 haploid, genetically unique cells, daughter cells, right? So this is 1, 2, 3, 4 cells. Each one of them is genetically unique. Each one of them is haploid. So those are things you definitely need to know for the exam. And it should make sense because, you know, if we're talking about mitosis, asexual reproduction, you want a perfect copy. So that should be the way to help you remember that it's, you know, they're genetically identical. In sexual reproduction, evolutionarily, the purpose of sexual reproduction is to give us diversity, right? Variety. So it makes sense that when you make your sperm or your egg, that you are making genetically unique cells. And we'll talk about exactly how genetically unique it is shortly. It's very fascinating, I think. Anyway, I won't say are there any questions about what I just said, but really quickly, I'm going to do what the book does and jump into a quick review of what we just said. At that point, I'll ask you if you have any questions. Bless you. We're both, all right, sorry. Back it up a second. Again, we're done talking about mitosis, we're done talking about meiosis. Now we're going to step back and take a big picture, look at these things, kind of compare and contrast them. For both mitosis and meiosis, the chromosomes duplicate only once, right? So we go through the cell cycle, and then right there, boom, in S phase, we duplicate our chromosomes, right? Only one time do we do that. Of course, in mitosis, after we've doubled our chromosomes, we cut it in half one time, right? So you double it, cut it in half, you're back at the original number. In meiosis, we double it the one time, we cut it in half, which brings us back to the original number, and then we cut it in half again, which brings us to half that number. So again, that's how you can remember that when you, with mitosis, you start with 1 cell, you end with 2. Meiosis, you start with 1 cell, you end with 4. Right? Even if you were to just draw it, like imagine, this is going to be so horrible. If you have 1 cell, you cut it in half, like you do in mitosis, hey, you got 2 cells. You cut it 4 times, you cut it in half again, and now you have 4 cells. This doesn't look like that, but maybe that's a good way to remember. Kind of looks like a scope to me. Anyway, any questions about this review? All right, the next slide will give us our next Latin's word, which will be, let's see, cells. Anyway, like we've already said, the events that are unique to meiosis occur in meiosis 1. Another way of saying that is that next bullet point, which is that meiosis 2 is virtually identical to mitosis, right? Because at that point, that's where we're separating the sister chromatids as opposed to the homologous chromosomes. And at that point, you know, meiosis 1 is where we have the crossing over. Right? We don't have that in meiosis 2. All those are going to be test questions. Which one is more like meiosis? Or excuse me, which one is more like mitosis? Meiosis 1 or meiosis 2? That's the test question. The answer is meiosis 2. Where does crossing over happen? Meiosis 1. Which one gives us haploid cells? Mitosis or meiosis? Meiosis. Which one gives us diploid cells? Mitosis. Anyway, any questions about this slide? Here's a picture of everything we've learned in one convenient picture. Again, we're dealing with the same parent cell. This would be a little bit more interesting and a little bit harder to follow, but I almost wish there was a textbook that had all 46 chromosomes instead of these hypothetical that have basically two sets of chromosomes, two pairs of chromosomes. Anyway, you can see here, you can compare prophase 1 to metaphase 1 to the anaphase, and then the anaphase 1 and all that. It's everything we just said. Again, in meiosis 1, we're separating homologous chromosomes. In meiosis 2, we are separating sister chromosomes. Any questions about that picture? All right, now let's talk about that question about the siblings. Let's really dive into that concept. The origins of genetic variation, and this is so, so important. We're giving you the origins of it now, and then the next chapter, when we talk about genetics, which is what this week's lab is about, as you guys already know, this is also important. Genetic variation will be important for genetics. Then when we talk about evolution, this is really, really important. Without genetic variation, there would be no evolution. As you already know, the offspring of sexual reproduction are genetically different from the parents and from one another. So like the question was asked earlier, is this why your siblings are similar but different? Basically, I'm paraphrasing the question. And yes, right? You are genetically similar to your parents, you're genetically similar to your siblings, but you are technically different, right? Unless you're an identical twin, in which case you are genetically identical to your sibling, but we'll talk about that later. So then the question is, how does meiosis produce this genetic variation? Again, this genetic variation is important moving forward. This is not a concept we're going to leave behind in this chapter. First thing we're talking about is independent assortment. It's very important. Make sure you're paying attention. It will get complicated. I think when I first tell you about independent assortment, you're like, okay, I've got that. That makes sense. But then later, I'm going to introduce another concept. It seems a lot like an independent assortment, and that's where it gets harder. Here we go. Independent assortment. When the chromosomes are aligned in metaphase 1, so picture it if you will, picture that picture I just showed you in your brain. They have all those homologous chromosomes lined up in the middle, because that's what happens in metaphase 1, right? They're all lined up. When they're lined up, the side-by-side orientation is by complete chance. What does that mean? Well, what I mean is, sorry for you guys online, you might not be able to see this. If you have, it's not like you know, when they all line up in the middle, this is what it looks like, right? 1, 2, 3, 4. This time I drew 4 pairs instead of the 2 pairs that you've seen in the textbook, and instead of the 23 pairs that we actually have. When they line up like this, it's not like this is all, they all line up like this. Mom on this side, and dad on the other. They don't do that. There is no rhyme or reason to the way they do that. That could happen. It is possible. And sometimes I'm sure it does. Where all moms are on one side and all dad are on the other. But what probably happens, excuse me, now I'm not going to draw the chromosomes. I'm just going to draw the M or the D, to represent the chromosome from your mom or your dad. What you get is maybe mom and dad on this side, mom and dad, maybe dad and mom, mom and dad, dad and mom, dad and mom, dad and mom, mom and dad, whatever. Imagine me doing that 43 times, or 23 times, excuse me. 6 chromosomes, 23 pairs. Imagine me doing that 23 times, and all the different ways I can put mom on one side and dad on the other. You guys see what I'm saying? The important thing here is when they line up, it's not all mom on one side and dad on the other, or it could be. But it's usually just completely random.
Could it be like mom, or dad, and mom, dad happen to be opposite?
Yeah, so if this is chromosome number 1, and that's chromosome number 2, and that's chromosome number 3, obviously for chromosome number 1, you're only getting one version from your mom and one from your dad, and they're all together. For chromosome number 2, you got one from your mom and one from your dad. Great question. So yeah, when they line up, it's completely random. So that's why, one of the reasons why you are different than your siblings. Even though you have the same, dipping from the same gene pool, so to speak, the version you get might be different, right? So let's assume, I'm not really going to make it simple here, simplify it. This is not the way it happens in nature because it's not this simple. Let's say they really are only 2 chromosome pairs. So chromosome number 1, I won't go that deep yet. Just know that, right? just know that when they line up, the moms and the dad chromosomes, they don't all do it mom on one side and dad on the other. That is what independent assortment is, and you need to know that. Here's where it gets interesting mathematically. For a species that has over 2 pairs of chromosomes, well, I already said that, never mind. For humans, since we have 23 pairs of chromosomes, there's a formula for this, if you will. I don't know if you want to call it a formula, it's pretty simple. But if we have 23 pairs of chromosomes, and there's 2 ways each one of those pairs can line up, that means to figure out the different number of combinations, you just do 2 to the 23rd. So that means there are 8 million possible combinations of sperm or egg, right? So when you produce your sperm and your egg, remember they cut in half, the possible combinations of which version of chromosome 1 did you get for your egg? Did you give the mom version of chromosome 1, or did you offer the dad version of chromosome number 2? Did you offer the mom version and the dad version? There's 6 or 8 million different ways for you to make that. Another way of looking at it is saying that is if I were to cut you open and look at your sperm or look at your eggs before I get arrested for cutting you open and doing that, I would find if I were to look at 8 million of your sperm or 8 million of your eggs, statistically none of them should be the same as far as which chromosomes they have. That's a lot. That's big numbers. Think about that. That's how unique you are. So that one sperm that you came from, there was 8 million different versions of ways you could have come out. That one egg that you came from, there was 8 million versions of which one you could have gotten. And then, mathematically, when you bring those 2 together, like what are the chances of you being that one sperm? What are the chances of you being that one egg? And then what are the chances of those 2 coming together to produce you? That's 1 in 64 trillion. So the chances of your parents would have to have 64 trillion offspring, statistically, to have another one identical to you. And I'm only talking about which chromosome versions you have. You know, for chromosome number 12, do you have mom's version or dad's version? Yeah, that gets a little bit cloudy, but anyway, that's the point, right? So that's a big thing for genetic variation. Your parents could have 64 trillion kids, and statistically, each one of them should be different based on which chromosomes they have. That make sense? I will say, I'll put it next to this, not because you don't need to know that, but because I'm not going to ask you those specific numbers. I don't remember if those questions are in the study guide or not. So if they're in the study guide, you need to know them, but you can just look them up. For the exam, I don't think I'll ask you that. Yeah, forget the X. Let me put a question mark. I don't know. I might ask you that. Anyway, are there any questions about that? Right. Now keep in mind too, we're still just talking about independent assortment. 88 million different versions, right? 64 different versions of children, right? If your parents were to have sex and have that many times and have children that many times. Here's what we're talking about. Here's what I tried to draw. Again, this is something that only has 2 pairs of chromosomes. This is an organism that has 2 pairs of chromosomes, unlike us. We have 23 pairs. So the possibility number 1 is, hey, look, all the mom chromosomes lined up on one side, all the dad chromosomes lined up on the other. So when this happens at the end, you have 4 gametes. You know, some of them have all mom versions, some of them have all dad versions. Here, they're lined up separately, and you can see where it's just different. The point here is, again, for independent assortment, they don't all line up like this, mom on one side, dad on the other. They line up randomly, independent of each other. There is no rhyme or reason. So any questions about independent assortment? All right, and that's not that complicated, right? That's a pretty simple concept. But like I said, later, I'm going to teach you a different concept. It sounds a lot like independent assortment. People get confused with independent assortment. So let that marinate and solidify in your brain for a while before we get to the other one. Anyway, the next thing we're going to talk about, because your book talks about it again, something we've already talked about, which is crossing over. So now we're going to talk about crossing over, but this time in the context of, oops, there we go. We'll talk about it in the context of sexual variety here. Anyway, as you already know, crossing over is the exchange of corresponding segments between homologous chromosomes, which is a fancy way of saying, when your homologous chromosomes get close to each other, sometimes they swap chunks, right? As you already know, this happens in prophase 1, not metaphase 1, not anaphase 1, not anything in meiosis 2, but prophase 1. And because of crossing over, gametes arise with chromosomes that are partially from mom and partially from dad. Meaning, again, like I already said, we saw how the mom chromosomes and dad chromosomes line up independently. But then you have to keep in mind, not only are they lining up independently, which you don't see that in this picture, but they're also swapping chunks, right? So here we have, a mom chromosome that has 2 chunks of dad on it, and a dad chromosome with 2 chunks of mom. So when that thing happens and they separate, you might get a fully mom chromosome, right? When we're talking about the eggs you're looking at, the sperm. It might have the full mom version, or it might have the mom version with the dad chunks, or it might have the dad version with the mom chunks, or it might be the full dad version. Again, I'll remind you, sometimes this doesn't happen at all. Sometimes, as we can see in this version, it happens twice. Sometimes, as we saw in the last version, it only happens once. Sometimes it's big chunks, sometimes it's small chunks. It's completely random. So keep that in mind. Not only are there 8 million different ways that your chromosomes can line up when you produce an egg or a sperm. Not only that, but then you take into account that even if you get a mom version or dad version, whatever, you might get the mom version with dad chunks on it, or the dad version with mom chunks. It's so much variety. So that 64 trillion is now blown out the window, right? Because like I said, for your parents to make you again, they would have to statistically have to have 64 trillion children, with one of them to be genetically unique or genetically identical to you. Then you throw this in there, then it's all out the window, right? Even if they had the same versions, like, all right, they got the same mom, dad version. But because of crossing over, even though they might have the mom version of chromosome number 7, they might have the mom version with dad chunks, and you might have the complete mom version. Anyway, any questions about crossing over? All right. Let's talk about what happens when things go wrong. Because remember, when we talked about mitosis, we said when things go wrong, that basically gives you cancer, right? Because, mitosis needs to start and stop at certain times. There's on-off switches, cellular on-off switches. And when those switches break, the thing is just constantly reproducing, and that's not good, and that's basically what gives you cancer. And now let's talk about what happens when meiosis goes wrong. And the short answer to that is, it can result in genetic abnormalities that range from mild to severe to fatal. Some of them, as a matter of fact, I'll tell you in a second. First, we'll talk about how accidents during meiosis can even alter the chromosome number. And it's something called non-disjunction. You need to know what non-disjunction is. Maybe because I'll ask it, I don't know if I will specifically or not, but definitely because I'm going to use this word a few times in this chapter. Non-disjunction is when the chromosome pairs fail to separate in anaphase. So I keep saying, right, you get those 23 pairs of chromosomes lined up in metaphase 1, and there's a tug of war, and they break, right? They separate. Well, they don't always separate. In that case, the homologous chromosomes don't separate. Or if we're talking about metaphase 2, then it's the sister chromatids that don't separate. Either way, they're not separating like they're supposed to. And when that happens, then you have problems, right? So in this case, we have non-disjunction in the first meiosis. So these homologous chromosomes right here failed to separate. So what happens is now this cell doesn't have enough chromosomes. This cell has too many. And then they go through another round of meiosis. So now we have 2 cells that don't have enough chromosomes, and we have 2 cells that have too many chromosomes. And then this version on the right-hand side, meiosis 1, it went just fine. The homologous chromosomes separated. But now the problem is when meiosis 2, when we're trying to separate the sister chromatids, in this case, the sister chromatids didn't separate for this cell. So this cell goes on and does its normal thing. So we do have 2 normal cells from that, but then this one produces 2 abnormal cells. Specifically, this one has not enough chromosomes, and that one has 1 too many. And that might seem trivial, and for some organisms it is. For some plants, that might be okay. But for humans, it can be bad. So for example, the next slide I'm going to show you is going to show you an abnormal gamete fertilizing a normal gamete. So, remember gametes are egg or sperm. And the result of what I'm about to show you is a zygote that has 2n plus 1 chromosomes. And as far as you're concerned, I'll put an X to that. I'm not even going to describe that. It just has an abnormal number of chromosomes. The fact that there's a plus in there, you just know that it has too many chromosomes. So this is how the thing started. Actually, I don't even need to read this. This is how it started, right? This is how it all began. For this person, there was a normal sperm that got together with an abnormal egg. This egg happened to have too many chromosomes. And when it got together, now this zygote, and we all started as a zygote. At one point in our life cycle, we were all just one cell. That one cell has one too many chromosomes. But the problem is not going to fix itself. Because how does that one cell grow into what we are right now? Mitosis, right? This thing's going to go through mitosis, which is going to make a perfect copy of this. So then we'll have 2 of these cells. Then I'll do it again, we'll have 4 of these cells. And then I'll do it again, we'll have 8, 16, 32, all of that, right? And it's always a perfect copy. So if you screw it up at the beginning, right, that's the master copy. If that's the master copy, and you keep copying that master copy, then all the copies are going to be messed up. So that's how it happens. Your book gives a specific example of when this happens, and it's called Down syndrome. I'm sure a lot of you have heard of it. Down syndrome is also called trisomy 21. You should know that for the exam, possibly. This kind of tells you what it is in the name. So chromosome number 21, as it is for all chromosomes, you should have one version of each, right? A mom version and a dad version. You should only have 2 chromosome number 1s, or 21s. You should only have 2 of any given chromosome. But in trisomy 21, you have 3 of them, right? You have 1 extra chromosome. You have 3 of the number 21 chromosome. So that results, obviously, in 47 total chromosomes instead of 46. The person who has trisomy 21 is said to have Down syndrome. The book gets pretty specific. I'm definitely not going to ask you these questions, but let's talk about it very quickly. It affects about 1 out of 850 children. It is the most common chromosome number abnormality. It is the most common serious birth defect in the United States. And the risk of this happening increases with the age of the mother. Specifically, 35 and up is kind of a dangerous place in the context of this conversation. So because of that, anybody who's 35 years or older are candidates for prenatal screenings. Again, I'm not going to ask you that. The important thing here is that you know the trisomy 21 is what basically causes Down syndrome, and it's when you have 1 extra 21 chromosome. 1 extra of the number 21 chromosome. This is a great thing to look up for independent work. What about the other 23 chromosomes? Well, we're going to talk about one of them, another one, but what about chromosome number 5? I'm just randomly throwing you that out there. What happens if you have an extra chromosome number 5, or an extra chromosome number 1, or an extra chromosome number 9, right? What happens if you have extra of any of them? Or what happens if you have too few? And I will say this, a lot of the times, this is such a bad, so drastic. Obviously, this isn't that drastic, because people with Down syndrome, they basically live, right, relatively normal lives. Not completely normal. They have short lifespan. You know, they look a little bit different. They have varying degrees of developmental delays. But generally speaking, they survive and thrive, right? But some of these are so bad that if you have extra chromosomes, it doesn't even make it, right? That's usually, I shouldn't say usually, that's a lot of miscarriages, or just because the chromosome numbers were off and it just won't make it. A lot of times people get pregnant and don't ever even know it, because again, extra chromosome or chromosomes missing, and the zygote is just so unviable, you just never, a lot of times people just don't even realize they were pregnant to begin with, because it comes and goes. So again, just an idea for independent work. Look that up. What about all these other extra chromosomes? So again, trisomy 21, see right here, on chromosome number 21, this is a person who does not have trisomy 21, right? So they only have 2 chromosomes. And again, to remind you, this is a carrier type. Here we are, this one right here, this person has 3 of those chromosomes. So what happens if you have any extra on any of those ones? I have a hypothesis. And I've been saying this for semesters and semesters, and I have yet to look it up. But you can see trisomy 21. These are the sex chromosomes. They're not that big. We're going to talk about them later. My hypothesis is the reason that you can survive having an extra chromosome number 21 is because look at the size of it, right? We're not geneticists. You guys aren't geneticists. But just by looking at the size of those chromosomes, you can probably tell there's probably not that many chromosomes or not that many genes on there, right? You guys aren't that familiar with DNA yet, but you can just tell by looking. That's not much DNA That's a lot of DNA, right? So this one probably has a lot of genes. That one probably doesn't have that many. So it's my hypothesis that you can afford a little mistake when you're only dealing with a few genes as opposed to when you're dealing with a lot of genes. It's just my hypothesis. I don't know if that's true, but maybe you can find the answer for it. Anyway, any questions about trisomy 21? All right, to piggyback on my hypothesis, next thing we're talking about is the sex chromosomes, which are not nearly as small as trisomy 20 or the 21st chromosome, but they're also not as big as some of the other ones. So we're still dealing with small chromosomes. And when you have an abnormal number of sex chromosomes, also again from disjunction, that's also usually not fatal. But it does upset the genetic balance, just not as much as the other stuff, right? I will say this, it's generally not deadly, right? So you can have an extra X chromosome or an extra Y chromosome, and it usually doesn't kill you. But there are some side effects. Your book goes into it a little bit. There's this chart right here. Next word for attendance, I'm going to circle it. So if you're watching this or listening to it, open your eyes and look what I'm circling. That is the next word for attendance. Anyway, you can see all these different syndromes, right? So this person has this male has an extra X chromosome. Remember, X is what makes the female. This male has an extra Y chromosome. This female has an extra X chromosome. This female only has one X chromosome, doesn't even have another one. So on and so forth. And you can look at these and read them and learn about them. I'm not going to test you about any of this. You can see how common they are. For criminal justice majors, this is slightly interesting, I think. I read a study one day where they were talking about this person, XYY. That's sometimes dubbed as super males, right? So the normal male is XY. This is the Y chromosome that makes you a male. So some males have an extra Y chromosome. And I read that in prison, the frequency is not 1 in 1,000 like it is for the rest of us, right? 1 out of every 1,000 people, or males, has that extra Y chromosome. But in prison, there's more of it. And I think the hypothesis is that if you have that extra Y chromosome, 1 in 2,000. Anyway, if you have that extra Y chromosome, then you produce more testosterone, and you're more aggressive, and therefore you're more likely to commit violent crimes. So it's something to look up if you're interested in that for independent work. Anyway, those are just some examples. And again, I'm not going into this because I'm not going to ask you about it on the exam, but if you want to, look it up for independent work. What exactly happens other than what I just told you about having an extra Y chromosome or an extra X chromosome or missing an X chromosome, right? You can look all that up. Look all those up if you want to see what see exactly how it affects you. Of course, it does list some of the symptoms right here, but if you want to dive deeper, you can. Any questions about the abnormalities of sex chromosomes in humans? All right. I always skip over this part, but I do want to say something this time. Every chapter ends with evolution connection. I skip over it usually, but this time it's important because this is important for the chapters that are coming up. Basically, we want to know what are the advantages of asexual reproduction versus sexual reproduction. Because remember, some organisms do both. Some of them only do one. If you're in an environment where you're just really thriving, right? You're just doing perfectly. You are perfect for that environment. And the environment's not changing. That's where you want asexual reproduction, right? You just want to make a perfect copy of yourself because you're doing a really great job. Or if you're just not around to have sex with anybody else, right? You're all by yourself somewhere. Obviously not you as a human, but as an organism. You're an organism and there's nothing else of your species around and you can't have sex. Obviously that would be an advantage to asexual reproduction. Why would we want sexual reproduction, which is what we do? Well, that's when we're not perfectly adapted to our environment and or the environment is changing, right? So it's good for us to have kids, genetically speaking and evolutionary speaking. It's good for us to have a lot of kids with a lot of different varieties because it ups the chance that one of them will survive. How many of you have heard of people dying of COVID? Hopefully all of you. You might not know someone specifically, I hope. But we all know people who died of COVID. How many of you know somebody who's had COVID and survived? Just a couple of you. I had it and survived, right? Point being is there's probably some sort of genetic differences where some survived and some didn't. And thank goodness for sexual reproduction because if we were all just that version, we just couldn't survive COVID and the whole human population would be dead right now, right? I'm simplifying things, but That's the point, right? We want a variety. So that'll be very important later in the semester. So that's it. I'll be online for office hours if you need me. If you want to take the pictures for the extra credit, we'll meet out in the hallway. And if you haven't done your pre-lab yet, go ahead and do it. It's better to get partial credit than no credit. And I will meet you guys all here at noon. Don't be late. Be here, please.